Well, we need to give John just a moment to get the recording going, and I think I've got that thing on. I don't know. Sometimes technology worries me. I'm afraid it's getting out of hand, and now they're talking about artificial intelligence, and that just scares me silly because it's already smarter than I am. But it's good to have the opportunity to present this second lesson in this series that we're entitling The Unanswered Questions of Science. And of course, this is leading up to our Vacation Bible School in June, which will deal with the subject of the week of creation. And so we're looking forward to studying that, and the kids always have a good time with that as we go through these 7 days of creation. And so we're really looking forward to our Vacation Bible School on June the 4th through the 8th. This particular lesson tonight is way longer. I'm going to have to go through stuff at a pace that's even faster than what I did at the last lesson. But I also taught biology and human anatomy for an advanced placement biology for 35 years in essence. And so there's a whole lot more material here than what I'll be able to get through in a reasonable amount of time, because nice to see it on the screen finally. As we talk about the unanswered questions of science, there's no doubt in my mind, after I consider myself a scientist back from ages ago. Since the time of Mr. Lucas at Martinsburg High School, really got me excited about the field of biology, about studying life. And since then, it's been a passion of mine. And so, without a doubt, in all of that almost 50 years now, well, more than 50 years pretty much, as I've studied nature, as I've studied the creation and looked at the answers of science, it was obvious to me, and I've said this before in lessons that I presented here, that when I was going through my undergraduate work at Shepherd University, I majored in biology and went 4 years, took all the evolution courses that I could work into my schedule and still graduate. And at the end of that 4 years, I thought, wait a minute, there's something wrong. I still don't see the answers that evolutionists keep saying they've got. And so I took another extra half a year of college, another semester of undergraduate work, and I took every evolution course that the science department offered, and it didn't help me any more to find the answers for the kinds of questions that evolution has to provide. The evolutionist science has to provide an answer. Of course, as we did in the last lesson, we want to start off with God's answer, because that's, of course, what I'm going to hang my hat on, because nothing in God's Word has been disproven by science. Now, there are unanswered questions, and everyone understands that. There are unanswered questions that as creationists, as people who believe God's Word, have not been provided. God didn't give the Bible to answer all of Dave Everson's questions. It's a good thing. It would have been huge, all right, because I've got plenty of them. But we see in Genesis chapter 1, and God made the beasts of the earth after his kind, and cattle after their kind, and everything that creepeth upon the earth after his kind, and God saw that it was good. Of course, we know that this is on day 5 of creation, but I wanted to focus on the fact that as we see in each of the days when God created living things, he created them and gave them the commandment to reproduce after their kind. And that's important, because that's exactly what we see in the world around us today. And it's exactly what we understand and what modern biology has come to understand. When elephants reproduce, we know that we get baby elephants. Pandas produce baby pandas. Butterflies produce butterflies. And plants, when, most of us are planting our gardens now, planting and then planting our gardens. And when we put a bean seed in the ground, we don't expect turnips to come up. We know turnips won't come up. If we put a bean seed in the ground, we're going to get beans. because those living things follow God's command to be fruitful and multiply after their kind. That's basic genetics. It's been basic genetics since they rejected Gregor Mendel's science studies with garden peas back in the 1700s. Modern science said that's garbage, that's worthless, ignore it. And of course, part of the problem with that was that Charles Darwin was doing his work and coming up with his models and answers of the evolution of life. And of course, Charles Darwin, his theories are still being challenged and researched because they can't find the answers. But Gregor Mendel's called the father of modern genetics because of the work that he did. He died not having a clue that he did anything important. That he thought his life as a scientist was completely worthless. But of course, we know that's not the case. And then, of course, we turn to the book of Job. And we know the story of Job and the problems that man went through. And we know that he questioned about what was going on in his life and why these things were happening to him. And we know at the end of the book of Job, in chapters 38 through 41, God says, okay, Job, I've had enough. Tighten your belt up because I'm going to ask you some questions. I'm going to give you a science test, and you answer me if you think you know so much. And of course, we know that Job wasn't, as we would be, able to answer anything that God asked him. But in Job chapter 12, verses 7 through 10, we see Job reveal a truth about the nature and what we can learn from it. When he said, "But now ask the beasts, and they will teach you, and the birds of the air, and they will tell you. Or speak to the earth, and it will teach you, and the fish of the sea will explain to you. Who among all these does not know that the hand of the Lord has done this, in whose hand is the life of every living thing, and the breath of all mankind?" And so we see that Job understood that when we look at the world around us, we can see the marvels of the creation, we can see the things that God has done, and we can understand because of what we can see. We can understand that God has this in his hand. And the first song that we sang, "Our God is Alive," verse 3, we sing that one on purpose when we have a lesson like this. "Secure is life from mortal mind. Though man can search, he's not going to find." We've searched for more than 200 years now, scientists have, and we're no closer to the answer than we were at the beginning of the search. And so, in what little time we have left, which is not, like I said, this could go on for a long time with the work that I used to spend weeks teaching this particular topic. Well, the question is, for evolutionists and for science, how did life begin? And I want to use some quotes from some scientists because, we have an answer. It says God did it. Okay, for in 6 days God created heaven and earth and all that in them is, in Exodus 20, verse 11 and 31, verse 17. And then in the book of Revelation, all things were created for thy pleasure. And so we can see. But when we look at the evolutionists, the science of the world's answer, the earth is beaming with life. And yet there is no consensus on how life arose, or, and I added the emphasis, or what life is. The origin of life is one of the great unsolved mysteries of science. This wasn't some unknown individual. This was Francis Crick of Watson and Crick, the discoverers of the structure of DNA, which we'll talk about in a little bit of depth here in a few minutes. But the questions that he asks, or the questions that he points out, how did life come about? And even what life is. Scientists have not been able to define what life is. When we started sending spaceships into satellites and things into outer space to Mars, they had to come up with a definition of, well, now, if we find something out there, when are we going to decide that it's actually alive or not? And they're no closer today. I just read an article 2 or 3 days ago as I've been background researching to make sure I had all my facts straight. But a study that was done and was written up just about 2 years ago said, and the title was, "Don't Tell Creationists This, but we still don't know what life is." That was the title of the article in Scientific American, not some no-name publication. This was Scientific American. They don't want creationists to know that they don't have the answer, because they've said, we have the answer. They've told us for hundreds of years, and it's in every biology book, oh yeah, we know how life came about, but they really haven't. And again, they're no closer. So, Francis Crick also wrote this in the same book, Life Itself, Its Origins and Nature, which he wrote in 1981. An honest man, armed with all the knowledge available to us now, and that has continued to today, could only state that in some sense, the origin of life appears to be at the moment to be almost a miracle. So many are the conditions which would have to have been met or have to have been satisfied to get it going. And so Francis Crick, and we'll see in a minute, he said it couldn't happen here on earth. There's no way it could happen here on earth. And he's come up with his own answer that we'll look at in just a minute. But he says it appears to have been a miracle. Bingo! It was a miracle. God brought it into existence out of nothing. I can't explain how God can do that. I can't explain what God is. The Bible only gives us information, and that's all we have to work from. And so we simply have to accept what he said, and we do have to have faith. Just like the evolutionist has to believe life is here, so somehow it got here, whether it was a miracle or whatever possible answer they could come up with. Well, a chemist wrote this. Most chemists believe, as I do, that life emerged spontaneously from mixtures of molecules in the prebiotic earth. How? I have no idea. We need a really good new idea. And then after writing about the prebiotic world and RNA transition and the RNA world and some of that, he said, I just don't understand how that works. Chemists have reached this conclusion because they have been trying to make life in a laboratory. Some of the best minds in the world for the past 200 years have been laboring at this, hoping to be able to create something that was alive, or to have life in a bottle and be able to pour it back into or onto something that does not have life and bring it to life. Of course, they're not capable of accomplishing that, and certainly never will be. Well, this individual that you see on the screen is Richard Dawkins. Wrote a book, Climbing Mount Improbable. Richard Dawkins is the world's leading evolutionary atheist, biologist. He doesn't believe in God, but when he's asked about how life began, his answer is nobody knows how it began. Nobody knows how it happened. And he says that the study of biology is the study of things that appear to be designed for a specific purpose. Well, that's exactly what God said. I'm going to create these things. They're all going to work together. Plants were created, animals were created, and they were designed so that animals give off carbon dioxide that plants need. Plants give off oxygen that animals need. And so when you abandon God as a possible answer, this is what we're left with. The questioning, the unknown answers that scientists have simply not been able to provide. Well, I mentioned that Francis Crick has studied this, studied DNA, studied the problems of how it could have come about and how life could have come about. And his answer, or one of his answers that he came up with shortly before his death, was directed panspermia. And this is sort of it in a nutshell. In 1972 paper, he co-authored with biologist Leslie Orgill, called Directed Panspermia. Crick suggested that perhaps extraterrestrials had seeded the earth with microbes sent in specialized spaceships that would protect the microbes on their journey. Folks, they're saying that aliens are the reason why life is here, because life evolved somewhere else in the universe. Remember when we looked at the unanswered questions of the universe, we talked about the problem of Where are all the aliens? Where's all the life out there? Life has had so much longer. The universe is supposedly 13 billion and a half years old. The earth is only 4.5 billion years old. Surely, in that period of time, life would have evolved somewhere else in the universe. And so Francis Crick said, that's where the DNA came from. That's where the beginning of life came from, was out there. Well, they haven't really solved their problem. All they did was move it somewhere else. And they assume that somewhere in the future, that whenever we're able to travel to Mars or travel to the more than 1,000 habitable possible planets outside of our solar system, that maybe we'll find the extraterrestrial life. We'll find the beginnings of life that they say, well, maybe DNA came on a meteor that hit the earth, and that's how life got started here on the earth. DNA evolved somewhere else and then came here. Of course, folks, that's a pretty weak answer. That doesn't really solve anything at all. It just creates more and more questions. Well, let's move on as we look into this questions of the unanswered questions of science from biology. How did complex molecules? All living things are made up of 4 basic atoms or elements: carbon, hydrogen, oxygen, and nitrogen. And I'll try not to get way down too far into the mud with this, but it gets pretty deep after a while. From those 4 atoms, we see that there is a plethora, a multitude, a huge number of complex molecules that have to come into existence. Things like carbohydrates, sugars, proteins, the things that make up enzymes and make up muscle tissue and make up so much of living things, and nucleic acids, which we're going to spend more time on, and then lipids, the things, the fats and the lipids the steroids are so essential to life itself. The question is, how did those complex molecules come into existence? How did they get started? From carbon, hydrogen, oxygen, and nitrogen, you can make carbohydrates, you can make proteins, you can make nucleic acids. Well, you have to add, the nitrogen, and you can make lipids. By putting those molecules in different arrangements, you can come up with large molecules called macromolecules or complex molecules. And the question is, how did those come about? Well, the question gets a little deeper, and we're just going to look at one possibility, one problem with this idea of these complex molecules. We could look at each one of them and spend, again, hours. The complex molecule carbohydrates. Sugar. I don't know about any of the rest of you, but when we go over next door, I'm going to look for the sugariest stuff I can find, because I'll burn up a lot of energy tonight and expended a lot of adrenaline. But I'll be looking for those sweets, the cakes and the chocolate and all that kind of stuff. All right? Those are made of sugars. The riboses, the sucrose, the glucose, the maltose, galactose, and so on and so on. Do you realize that science has not yet come up with any explanation for how those complex carbohydrates, like beta-ribose and alpha-deoxyribose, which are part of the DNA molecule, which we're going to talk about in a minute, how those came into existence? A recent research project, and the answer that they gave in their write-up was that sugars came from formaldehyde. Most of you may not know what formaldehyde is, but that's the stuff we use to pickle dead frogs. Okay? It's lethal if consumed, and somehow they believe that sugars, that nice sweet stuff that's in chocolate and cake and candy, and came from formaldehyde. That's the best answer at this point. And folks, there's a huge difference between the molecular formula of formaldehyde and even the most simple sugars, the glucose, the sucrose, etc. Big difference in their molecular formulas. And so the question is, we don't know how even sugar came about, let alone the lipids, let alone the nucleic acids, let alone the nitrogen-carrying bases. Well, let's move on quickly. Time's running fast. We're going to look at DNA, deoxyribose nucleic acid. We're going to look at 5 different aspects of it. because each one of them, there has to be an answer for how this macromolecule, how this huge molecule could do what it's capable of doing. Because in the very basic sense, this is insane that this molecule is capable of doing this. Well, what is it? DNA is a self-replicating material that's in every living organism. In simplest terms, it is a character of all genetic information. It contains the instructions needed for organisms to develop, grow, survive, and reproduce. It's one long molecule that contains our genetic code or our recipe. The recipe for all living things is found inside of its DNA. That's the abbreviation for deoxyribonucleic acid. And I've used this example before. I have a hitchhiker's thumb. I have it because my DNA tells me I'll have it. You don't have it because your DNA tells you to have a straight thumb. And every characteristic of each one of us that makes us, I'm glad you all don't look like me, and I'm sure you are too. All right? We all look different, but we're all human beings. It's because of our DNA. We don't look like an elephant because we have DNA that's different, arranged with a different recipe. And so DNA is important stuff. It's valuable to life as we know it. Otherwise, all life, if they all had the same DNA, would all look the same. Everything, if we had the same set of genetic code, or the same recipe for how we were to be made and how we would look, we would all come out exactly the same. Fortunately, that's not the case. This is roughly what DNA looks like. Watson and Crick in the early 50s came up with basically the pattern for what DNA is. It's a double chain molecule. These are the rungs of the ladder. Sometimes they call it a ladder. And you can see that it's all twisted up, but it has rungs which has a sugar here and a phosphate molecule here. Another sugar, another phosphate, etc. Same thing down this side, except you have a 3 end up here, which refers to the position of the carbon and the hooking, the co-joining atom, and a 3 end down here running in the reverse direction. So you have a 3 end and a 5 end of the DNA molecule, but in between is where the code is. This is where the recipe, folks, A, T, C, and G, adenine, guanine, cytosine, and thymine, are the recipe for why I have a hitchhiker's thumb. That's the recipe. That's the code that tells me I'm going to be 6'6", and some of you can only hope to be, taller than 5'9", John. No, all right. But it's in our recipe, folks, and it's in how these letters are arranged. And you can see those letters going up through here. Make up our recipe. And so that's the basic information about DNA. It is a molecule, macromolecule, that's present in every cell of the body. It's in, and I'd have to back up a little bit if I'm being true to my human biology, my human anatomy, I have to say that there's one cell that doesn't have DNA as an adult cell, and that is your red blood cells. As your red blood cells form in order to carry more oxygen, have more of the hemoglobin molecule, the DNA is pinched out of the red blood cells, the erythrocytes, and that way you can carry more oxygen. So the red blood cells, adult red blood cells, don't have DNA, but when they're in their formation process, they do have DNA. But it's found in every cell in the human body and in every cell of every living thing, from the single-celled organism all the way up to the giant redwoods, the blue whale, etc., etc. The strands of DNA in one cell are about 20 inches long, all right? But squeezed into the space of about 0.09 micrometers, it would take 40,000 strands of DNA laid down side by side to equal the thickness of a human hair. 40,000 DNA strands to make the thickness of a single human hair. This is an extremely small macromolecule, but it's extraordinarily important and certainly extraordinarily powerful. Well, how big is it? If you unravel the DNA in all of your cells, about 75 to 100 trillion cells is about what adult humans have. It would stretch for, and lay them down end to end, stretch for about 10 billion miles from here to Pluto and back. All right, that's a lot of DNA running around in your body, folks. I mean, we're talking about a huge amount of this macromolecule. Interestingly enough, it's extremely long if you were to stretch it out, but as we'll see in just a couple minutes, it's extremely small because it has to fit in every cell in your body. Well, how does it do that? How does that DNA, which about 20 inches long when you stretch it out, this is what it looks like whenever you see a chromosome, which is one of the 46 parts of the DNA that's found in the nucleus of each cell. This is just one chromosome, and these are sister chromatids, and they're attached to a central point called a centromere, and I'll not bore you with all that. Travis had to pass that test once upon a time. But the DNA undergoes something called packing. In order for DNA to be found inside the nucleus of every cell of our body and every cell of every plant and every bacteria, it has to be packed into a certain condition. Chromosomes, DNA doesn't always look like that. During the regular part of cell functions, your DNA just looks like a bunch of spaghetti in there. Just looks like a plate full of spaghetti, because it's all just sort of mixed up, and that's when it's working. That's when it's doing what its function is to do. But whenever it's getting ready to undergo the process of cell reproduction or copying itself, because remember that all of us, every living thing starts out as one cell. One single cell, and from that one single cell, we divide, become 2, then 4, then 8, 16, 32, well, you do the math. John can quote you all those numbers if you'd like for him to. Just keep doubling them. All right? And so that's when your chromosomes will look like that. When they're getting ready, as we'll mention in a few minutes, undergo mitosis. But in order to take that DNA and to make it look like this, a chromosome in what's called metaphase, in the 4 steps of mitosis, it has to undergo a organization, a packing process. And it has to be packaged a certain way because your cells have to be able to find the part of the instructions, the part of the recipe that it happens to need at a particular time. I can tell you tonight and tomorrow, Dave Everson is going to need to replace a lot of adrenaline. I'm flying high right now on adrenaline. Okay? I'm going to have to replace that molecule, those molecules that I'm burning up right now. And that's why I'm going to go eat some sugar too, because I'm burning large amounts of sugar. All right? The information for how to make adrenaline is in my recipe, is in the DNA, but it's only in a certain section. And in order to get that information out, the little man in there, and I'll refer to the little man in ourselves because there's got to be something in there directing all this. Otherwise it wouldn't happen. It's got to go to a certain part of the DNA and access it. Well, there are special proteins called histones. They're little round BB-looking molecules, and the DNA strand, the double helix strand, wraps itself around those, organizes itself around those, and then it wraps around again, and then it becomes basically, and again I'll date myself, one of the old phone cords that plugged into the wall, plugged into the phone, and it was all coiled up. Okay, these young punks don't know anything about that. They've never seen one of those phones. But that is how it fits into this metaphase chromosome, and still is such a huge molecule. It's called DNA packing. Now, can you imagine the first cell that came into existence by purely accidental processes? Imagine this process, that histones had to be present, the RNA, or excuse me, the DNA strands had to be present with their their sugar, their phosphate, and their 4 nitrogen-carrying bases. They had to be in there in the exact blueprint, in the exact order, in order for that first single-celled organism to come into existence, that bacterium, which they believe is because it's the simplest, okay, even though it's got 5,000 chromosomes, it's still the simplest life form that we know of. All of this had to be present at the very beginning. There is no known method by which any of this could happen without all of that being present and functional. And we'll see one of the biologists quoted here in just a minute. Well, DNA has information in it, obviously. It's got the blueprint. It's got the recipe. All right? A single gram of DNA is capable of holding an amazing 700 terabytes. You can ask Travis. I checked this 2 or 3 times, Travis. Harvard University biologists were the ones that were quoting this number. 700 terabytes is what a single gram of DNA it could hold. If we wanted to store all the digital information in the world, all we would need would be a teaspoon of DNA. That's all of the information, all the digital stuff that's in the entire world would fit in a teaspoon of DNA. Matter of fact, computer experts are trying to figure out how to get DNA to store, how to program DNA to make it store the information we want. Because if it did, back in the late 60s when I worked for the IRS, the computers were massive. They were mainframe. They used great big rolls of magnetic tape, 1-inch rolls of magnetic tape, and they covered just a massive area. The same computer now, my phone's down there, but the phones that we carry around with us have more computing ability than that mainframe IBM computer. If they can ever get DNA to store digital data and write it and rewrite it and unwrite it, etc., we'll be able to hold all the world's computers in the size of a teaspoon. Everything, all the information in the world, all the computing ability in the entire world would fit in a teaspoon. Well, Michael Denton said this: DNA is 2/millionth of a millimeter wide. A teaspoon of DNA could code proteins for all species that have ever lived and still have room for the information in every book ever written. DNA, the information that it has and that it can contain, is a staggering amount. Human DNA is like a computer program, but far, far more advanced than any software we have ever created. And this was Carl Sagan wrote this in the book The Road Ahead in page 228. And that is absolutely true. DNA is a computer program. If we nailed Travis down back there, and he'll tell you he's a computer programmer. If you put garbage in, Travis, what comes out? Garbage. Garbage in, garbage out. You have to know what you're putting in to get something useful out. I couldn't do that. Travis can. I've seen his work several times. All right? But it takes an intelligence. It takes knowledge to be able to program the computer. And the question is, how did the information get in the DNA? By an accident, the first time. The information content of a single cell has been estimated to be around 1,012 bits, comparable to about 100 million pages of the Encyclopedia Britannica. And this was Bill Gates. You might have heard of him. Bill Gates, Microsoft, all that kind of stuff. And yet he says 100 million pages of the Encyclopedia Britannica would fit inside the DNA of one cell. One cell. Where does the information come from to how to make? And you pick whatever plant you're seeing flowering now, whatever animals you're seeing, every one of them had to have that information available to them. So every plant, every animal, every fungus, every protozoan, every living thing had to have DNA, and that DNA had to be coded. It had to have the right recipe. Because if you get the wrong codes in here, the A's and the T's and the C's and the G's and all that, if you get them messed up, well, garbage in, garbage out. You end up with something that isn't what you wanted, and we call it a mutation. And of course, that's never been a good thing. Well, so DNA, how it came about, its origin, its information, from the government website, the NIH, Institute of National Health. The invention of DNA required the appearance of enzymatic activities for both synthesis of DNA precursors, retrotranscription of RNA templates, and replication of single and double-strand DNA molecules. In order to get DNA the first time, you had to have all these things present. This one I particularly like. Here at the molecular level are the roots of the old puzzle about the chicken or the egg. Which came first, function or information? As we shall show, and this Nobel laureate went on to explain, as we shall show, neither one could proceed without the other. They had to evolve together. So DNA had to be functional at the very beginning. DNA had to have the information so that it could be functional. And it couldn't be functional without, it couldn't have the information without its function being, his function working. It's the chicken and the egg, only it's a whole lot smaller. All right? And it's not, chicken came first if you're a creationist. God created a chicken, an adult chicken. Just like he created an adult man, said, be fruitful and multiply. You couldn't tell a baby that. All right? But you could tell an adult that. But this problem of DNA, which came first, both of those, the information and the function, had to come about at the same time, and boy, are we in trouble. All right, DNA carries out what we call mitosis. Mitosis is simply the process of cell division. Babies start out one cell and then divide. They're born. They don't stay babies. We got Oliver back there, and Guy and Abigail are probably napping, right, in the nursery. But between 2 and 10 trillion times a day, our cells undergo mitosis. That's a lot. This is what it looks like. Prophase, metaphase, anaphase, and telophase. Those are your chromosomes. This is what chromosomes normally look like. That's what your DNA normally looks like whenever they're in there doing its thing. And then before they're ready to divide, they have to come together into their component parts. There are 46 individual units, so that the process, you can take half of them, one side from each of them, and put it in what's going to become the 2 new cells. Folks, that has to happen, and it happens a lot, between 2 and 10 million times. We don't have time to go through all this, but this is what happens. It is exceptionally complex. The strand of DNA will actually come undone. It's unzippered by an enzyme called helicase, can't even say it now. And then molecules called DNA polymerase will fit onto the 2 strands, the daughter strands that they're called, and then it will start bringing in from outside of the nucleus the parts that it happens to need to make an identical copy. So that if you had A on this side, you'd have T on this side. If you had C on that side, you'd have G on the other side. And starts building that DNA recipe. Starts copying it. And folks, this is crazy, all right? DNA is a fragile molecule. On average, the DNA in a simple cell is damaged between 1,000 and 1 million times every day. DNA is a very fragile molecule. It's easily damaged. It's easily messed up as this process occurs. So there has to be some method by which these errors are fixed. And the story really gets deep now. Because you have specialized parts of this DNA polymerase protein. This is a protein that is responsible for making a copy of that strand, the template strand, the single strand of DNA, and coming out with a complete strand on the other side. All right, so it's showing here that there's a base, one of the nitrogen-carrying bases here, that's the wrong one, but it was put in by mistake. And there's a problem with that, and that has to do with molecular size, and you get mismatch, and you get all kinds of strange things happening. But on that DNA polymerase, there's a special domain called the exon nuclease domain. And folks, it's down here. Somehow this protein, this complex macromolecule, says, wait a minute, time out. you got it wrong. You got the wrong piece in there. You know, the mechanics didn't know what they were doing whenever they put the parts back together. They got the wrong piece in there. Now, tell me how this little portion of that DNA polymerase knows what's right and what's wrong. It's not been answered, and scientists haven't even got close to figuring that one out. Well, what it does is it says, okay, we're going to fix that. It brings in the correct base and puts it in, takes the old one out, puts that in, and we now have a correct DNA sequence. How did DNA learn to repair itself? Learn to repair the mistakes that happen. And folks, I will tell you, we know what the result is of DNA that does not get corrected. It's called cancer. That's the cause of cancer. DNA that is not copied correctly starts to have the wrong information, the wrong recipe, and the recipe, instead of saying, well, your mitosis sequence is only going, it should take you about 2 to 4 hours, it's going to happen in 2 to 4 minutes. And so one cancer cell, one cell that has the recipe wrong, then can start multiplying. We know that cancer grows in some individuals, in some cancers, at astronomical rates. Saw a patient, saw the surgery, removed a cancer weighing 56 pounds. Less than 2 months before the gentleman had had a doctor's appointment, there was no cancer there. In 2 months, it went from nothing, it may have been there, but the doctors couldn't detect it, to a 56-pound tumor. Took 4 doctors to lift it out of his abdomen. All right? That's what happens when DNA doesn't copy itself correctly. And of course, we know that it needs. Well, whenever we get to the process of producing offspring, which all living things, God commanded, be fruitful and multiply after your kind. And so all living things have to undergo the process of meiosis, or gamete formation. And in this process, humans are to have 46 chromosomes in each one of our cells. If dad gave 46 chromosomes and mom gave 46 chromosomes, it's 92, right, John? I think that's the math. 92 chromosomes. That wouldn't be human anymore. And so what has to happen before dad sends his genetic confirmation, his part, okay, it has to be reduced from 46 to 23, just 1/2. Mom does the same thing, 46 down to 23, okay, their complement then can, when they go together, become the 46 normal genotype. And this process is under what's the process of meiosis. Meiosis is a very challenging process because it happens in 2 phases, meiosis 1 and meiosis 2. But the end result is not a normal body cell. These cells have half the number of chromosomes, and in this case, especially in the case of the male of all species, they will have tetrads. They'll have 4 cells that will be produced. The female complement in this will produce 1 egg cell and 3 polar bodies that are not functional for reproduction. And so this process, how did this information, how is this carried? How was this put into the DNA the first time in order for this to happen? And this happens in all living organisms. This process of DNA reduction in gamete formation. Well, then if we take the male and the female, they each have their component. They're 1N, which means they're half the normal number of chromosomes. When they get inside of the egg cell, then it's quite the event. An event that does not have scientific explanation. How those 2 sets of chromosomes find where their corresponding part, chromosome 1, chromosome 2, chromosome 3, trisomy 21, for example, is when dad's chromosome 21 has 2 parts to it instead of the normal 1. And so whenever it combines in the egg, in the embryo, the zygote, 21 has 3 chromosomes instead of the normal 2. The recipe's wrong, all right? And that's not healthy. The baby is not going to be a normally developed individual. And so this process of zygote formation, of the combining of this DNA into the proper recipe is vitally important. Well, let's talk about another type of DNA. It's called mitochondrial DNA, and all I'm going to say about this is it's found in a cell organelle called your mitochondria. It only comes from the mother. All right? As this egg develops, mitochondria from the mother passed into the egg cell, and then from there to the baby. All right? They have now traced the genetic makeup of mitochondrial DNA back to 3 original mothers. Mitochondrial DNA, by doing the standard DNA analysis, there were, at one point in human history, there were 3, Ham's wife, Shem's wife, and Japheth's wife, Noah's sons, who received the command after coming off the ark to replenish the world. And science has proven just exactly what God said would happen. Well, as we wrap this up, well, there's another 4 hours right here. Cell organelles, how did they come about? We're not going to use 4 hours, believe me. These are the parts, the cell organelles, the parts of the cell that have to be present. You have the nucleus, of course, which contains the DNA. You have the endoplasmic reticulum, which is the transportation system in the cell. You have the Golgi apparatus, which is involved in packaging up Dave's adrenaline that I used to be released whenever I started talking tonight, and I started breaking open little vesicles of adrenaline that started flowing into my bloodstream, and now I can't stop talking real fast now. All right, but that's the Golgi apparatus. Okay, mitochondria, that's the powerhouse of the cell. Centrosome, involved in the division of each cell during the process of mitosis, ribosomes, lysosomes. Those are your police force, by the way. These are your safety guys. Well, I want to talk about one, and then the lesson will be yours. This is called the kinesin motor molecule. All right? They're called motor proteins. This is a protein this is a product of the recipe of the DNA that has been produced. It's been produced outside of the nucleus. The blueprint, the recipe was brought out of the nucleus for how to make a particular molecule. Might have been the adrenaline molecule. We'll use that. That's what it is. But from where it was produced to where it needs to be on the outer border of the cell, well, it's a long ways away in terms of the cell. How does it go from inside the cell near the nucleus to the outside, which is like going from here to Charleston? Well, if we wanted to move something large from here to Charleston, Scott can tell us how that's done. Put it on a truck, don't you, Scott? And you drive it down the road. Guess what? This kinesin motor molecule does exactly that. It will attach to the molecule to be moved. This is what's called a microtubule, which is part of the endoplasmic reticulum framework running throughout the cell. And yes, it moves. It drags this molecule along. They've actually got electron photomicrographs of this happening. And these 2 little protein structures down here are like little feet. And they pick them up, they release, and they walk. And they walk down this microtubule carrying this adrenaline molecule for Dave so that it's out there ready to be released next time he preaches a sermon or the next time he gets scared real bad. All right? How did these structures come about? By accident. Well, of course, God answered it. The psalmist answered it in Psalms 139, verse 14. I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works, and that my soul knoweth right well. And if David, the psalmist, could say this about 4,000 years ago, can you imagine what we would have to say, given what we know about the complexities of the human body and of a bacterium's structure and the DNA and the chromosomes and all of these micromolecules and cell organelles? What we know now is so much greater than what the psalmist David said. what he knew at the time? Well, there are no answered questions with God. Certainly there are questions Dave would like to have answered, but we have everything we need to know to be pleasing to him. The Bible provides the answer. God planned and controls every function in every cell of our body. I got news for you. Dave Everson can't keep track of where he needs to go tomorrow, let alone how to operate every little thing that's going on inside of my body. Somebody's in there directing the whole show. Well, we've never found a little body, a little man in there running the whole show. All right? God's in control. God is control. Though man may search, he cannot find, for God alone does understand. God knows what's going on inside ourselves. God planned the method for our existence. Of course, he also planned our salvation. He left us the plan to hear, believe, repent, confess, and be baptized. And certainly that is the method by which we can be acceptable to God. I hope someday to have the answers to all these questions that I have that God hasn't told me the answers to. He hasn't provided everything I need to know, but he has given me everything that I need for life and godliness. This evening, the question is, what will your answer be? God has an answer for how you can be pleasing to him. The question is, what's your answer going to be to him tonight? If you need to obey the gospel plan of salvation, please let that be known. If you've done that and you've fallen back into the world and been tried and tempted by Satan as Job was and given in, you can make those things right. So whatever your need might be, we'd encourage you to come while we stand and sing the song selected.